Yeah, chitin plays this structural role in the cell wall of fungi. But what might not always be so obvious is that we can have smaller versions of chitin called chitin oligomers, and that these chitin oligomers play an important role in signaling. And one of my favorite chitin oligomers is called the lipocyto-oligosaccharide. Like the name suggests, it is a chitin oligomer with a lipid chain attached. And what makes these really cool molecules is that we used to think that they were just exclusively made by symbiotic microbes. So the idea was that these microbes would secrete this molecule into the soil and basically have a chemical exchange with their symbiotic plant partner, initiate some sort of plant response. So in the example to the right, we're looking at mycorrhizal fungi colonizing the roots of a plant host. And before I continue, I wanted to give you a closer look about how a LCO looks like. I'm going to be calling lipocyto-oligosaccharide an LCO. So we have the chitin backbone and the lipid chain. That's basically what an LCO looks like. And we can have variability here. We can have differences in how long the chitin backbone is, and we can also have different structures in the lipid. And we also have functional groups that can decorate the molecule. And this is pretty important, especially for plants where it's important for host specificity. And I also wanted to give you a brief crash course into the nomenclature. So here is basically how we formally refer to an LCO molecule. What this is telling me is that this is an LCO with a chitin degree of polymerization of 4. So 1, 2, 3, 4. Then things in the parentheses, we start with the structure of the lipid, which is a palmitic acid. This is followed by the functional groups, which in this case, it's the sulfate group. So, So one of the things that we find is that LCOs are not produced by simply microbes that are symbiotic. They're produced by most fungi throughout the fungal kingdom. And one of the questions that a previous graduate student, Dr. Tomas Allen Rush now, he asked, do fungi respond to these molecules? So the model that he uses is called Aspergillus fumigatus. And Aspergillus is a saprophytic organism, which means that it eats decaying matter, but under the right circumstances, it can become a pathogen to humans. And what makes this a really popular model is that first, it produces LCOs, and second, it's very genetically amenable. So I think just last year, they came out with an entire gene knockout mutant collection. So it's a pretty good resources when you're working with filamentous fungi. So Dr. Rush decided to ask if LCOs are so popular throughout the fungal kingdom, what are they doing? So he treats Aspergillus with LCOs and looks at what phenotypes occur. And the first thing that he looks at is germination of spores. So here we're looking at 4 different LCO molecules that he tested. He also tested things that would theoretically make up an LCO molecule, so just different chitin oligomer fragments of different sizes, and also just different lipid structures. And what we see here is that a sulfated LCO with a palmitic acid tended to increase spore germination. Though this wasn't necessarily just specific to LCOs, because oleic acid did something similar. But something that was specific to LCOs was how it changed the morphology of hyphae. So what we're looking at here is Aspergillus fumigatus, I want to say around 12 to 13 hours after germination. So here we have that the main hyphae of that individual. And what we're looking at here are the secondary branches. And what we see, and these branches are important for a lot of things, but a big one is how it colonizes. how it colonizes its environment. And that same molecule that seemed to increase germination, now we see it decrease the number of secondary branches. And this seemed to be specific to this LCO molecule. And during my time here, I, alongside an undergraduate researcher, Aidan Schmidt, we decided to go into this rabbit hole of looking at biofilms in Aspergillus fumigatus. Basically, they follow the same idea that they do in bacteria. They have a unique extracellular matrix. They have an increased tolerance to antimicrobials, and they also have more adherence to surfaces. So we decide to basically treat the fungus with LCOs and the fragments that would make up an LCO, and then do a crystal violet assay. So here is that data. We have the control. We have that LCO, that sulfated LCO with the palmitic acid, and just the fragments that would make this molecule up, the chitin oligomer and the palmitic acid. And we see that the LCO tended to decrease the optical density. And this optical density we use as an approximation for biofilm formation. What I'm concluding here is that this is LCOs are reducing biofilm formation. So that kind of leads to, and from now on, when I'm saying an LCO, I'm going to refer to this specific molecule. I just don't want to say the entire name of the structure every time I talk about it. So what we see here is there's this regulatory molecule that is common throughout the fungal kingdom, not just produced by symbiotic microbes. And we hypothesize that filamentous fungi are secreting this into the extracellular matrix, which they can then take up to basically do self-signaling and basically regulate different phenotypes involving development. But because they're being secreted into the soil, we also hypothesize that they're important for non-self signaling. So other individuals can take it up and basically have their own development altered in a form of crosstalk. So this sort of leads to one of my bigger questions, which is how do they recognize and respond to LCOs? Specifically, I can narrow this down into two big questions. Uncover the genetic pathway involved in how Aspergillus responds to LCOs, and also find the mechanism by which Aspergillus perceives LCOs. So let's start with that first one. So Dr. Rush, who did all that research discovering that LCOs were common throughout the fungal kingdom, he also left us with an RNA-seq experiment where we have Aspergillus fumigatus 30 minutes and 2 hours after treatment with LCOs. And here we see a large number of differentially expressed genes. And my idea here was, okay, I have this list of differentially expressed genes. If I want to see what genes are important for LCO response, all I have to do is look at the big regulators, so the transcription factors, knock them out and see which one stops responding to LCOs. But that seemed very an easy idea in the beginning, but then I realized that there's more than 30 transcription factors, and that might have been a very inefficient way to go about it. So what I was thinking then is like, maybe I could take a systems approach. So if I had, let's say, a regulatory gene network in Aspergillus fumigatus, where we're looking at gene-gene interactions, except that we're adding directionality. So we're saying that this is a target and this is a regulator. Maybe I can look at that list of differentially expressed genes, map it into the regulatory gene network for Aspergillus fumigatus, and maybe it'll map into a specific part of the network. And if it does, I can look at the main regulator of that part of the network and basically hypothesize that this regulator is important for response to LCOs. So in comes Dr. Shushmita Roy from Over the Wid, and Shushmita Roy has this really cool program called Merlin, which is able to infer regulatory gene networks based on the expression data for an organism. And what it requires is fundamentally two things. All the gene expression data. So in our case, it was all the RNA-seq data sets for Aspergillus that we could find. In this case, it was 18, and a list of regulators. So in our case, it was all the transcription factors, all the signaling proteins that we knew about in the fungus, and any gene that had the Go term enrichment annotation of development, regulator of gene development. And we put that into Merlin, and we get out is a very colorless network, apparently. We get a regulatory gene network that's then subdivided into what we call modules, where within each module we have gene-gene interactions, where we're looking at where some genes are assigned as regulators and other genes are assigned as targets. And here, all the regulators are assigned as regulators because we previously told them what is expected to be a regulator. So after doing this, the people at the Roy lab were amazing, and they basically whip out this large regulatory gene network for Aspergillus fumigatus. This is all the predicted gene-gene interactions, where the little red splotches represent major regulatory hubs. And in summary, this network can be broken down into 158 network modules. So now that we have this network, I am asking myself, okay, do those differentially expressed genes that I initially saw, do they cluster in a specific module? And the answer was yes. So several clusters or modules, the name is interchangeable, they were enriched for those differentially expressed genes, and I did what I did best and just looked at the top hit. That was cluster 5227, and what that looks like visually is this. So what we have here at the top are the gene IDs of all the targets in the module, and on the side we have all the regulators, and here this color represents who's targeting who. So what I immediately notice here is that we have a big regulator. So this one is regulating all these genes right here. And that regulator is a transcription factor known as ATFA, which has been pretty well characterized in Aspergillus fumigatus. More importantly, it was initially discovered to be important for asexual germination and the stress tolerance of that asexual spore. But more importantly, it seems to be regulating a lot of my differentially expressed genes. So I'm asking myself, is this transcription factor important for LCO response? And that would have been a really interesting thing to look at, because in my list of transcription factors, that more than 30 transcription factors, it wasn't one of them. Just because even though HFA is on the list of differentially expressed genes because it's significant, it's not very highly expressed, so it wasn't that interesting. Yeah, and those are the genes that were mapped to this module. So I acquire mutants, a deletion mutant of ATFA and an overexpression ATFA on the right side compared to the wild type. And what I see is that the wild type here, we have a healthy hyphae, but in the deletion, we have like a little squiggly phenotype with a lot of branches. And in the overexpression ATFA, we have like a little noodle that's really long with little to no branches. So I'm asking, they seem to have an important role in the phenotypes of hyphae. Do they respond to LCOs? And the answer is yes. I mean, the answer is they don't respond to LCOs. And here we're looking at secondary branches per micrometer. We have 3 strains, the wild type, the deletion and overexpression ATFA, and our 2 treatments as a control on the left, and LCOs on the right. In the wild type, we see that expected reduction in secondary branches, but in the deletion ATFA and overexpression ATFA, that seems to be gone. This is telling me that this transcription factor is important for the response to LCOs. So I feel like here, like I added a piece to the puzzle about what I think is happening. If we zoom in on what's happening at the cellular level, I hypothesize that LCOs are somehow being recognized by the fungus, and it's leading to a cascade of events that inevitably leads to the activation of ATFA and subsequently are phenotypes. So what I'm doing next is basically deciding to go upstream. So what is that signaling cascade? Here, the literature that's already been done on ATFA, it already has characterized in saying that ATFA is activated by the MAP kinase pathway, and upstream of that is the two-component system. And this is recapitulated by what Merlin is saying, because those enriched clusters are also have regulators with regulators from the two-component system and MAP kinase pathway. So that was good to hear. So that's what I'm tackling next. The next thing the next part of the story is basically finding how Aspergillus senses LCOs. So here, Merlin wasn't really able to offer me a good candidate, but I feel here like we can learn a lot about how plants recognize LCOs. So again, plants need to recognize LCOs to initiate some sort of symbiotic association. And it does this through a type of transmembrane receptor, one that has a small domain called the lysam domain. And this isn't just necessarily just for LCOs. This is just to recognize any chitin fragment or things that look like chitin, like peptidoglycan. Like basically, it basically sandwiches the molecule together and initiates some sort of response. So I'm thinking to myself, like, okay, maybe a lysam domain receptor is what's binding LCOs and initiating our cascade of events in Aspergillus fumigatus. So in the fungus, there are 8 predicted lysam domain containing proteins. And I was hoping that there would be one that's a transmembrane domain, because in my head, that's what I think a receptor is. But unfortunately, most of them are intracellular chitinases. And I was thinking to myself, an LCO is coming from the outside, so I would be thinking that it'd be something that has to sense the outside. But there were 2 effectors, and these effectors are secreted proteins with no catalytic domain. Lysam effectors have been shown to be important for phytopathogens, so pathogens of plants, because it's able to sequester chitin fragments and not let the plant be able to capture those chitin fragments to initiate some sort of immune response. And a paper recently characterized these 2 effectors in Aspergillus fumigatus. They tagged it with GFP to see where they localized. And while LDPB seemed to be localized to the cross walls of the fungus and something that we call a septa, LDPA, the other lysam effector, seems to be localized to the cell wall. So I was thinking, okay, it's not necessarily being secreted into, let's say, the soil, where there's no way it can really get back into the cell to signal. But if it's at the cell wall, it can maybe perceive an LCO and somehow relay that information back into the cell. So I acquire these mutants and I ask, are they important for LCO response? So here I have 4 different strains. I have the wild type double deletion of those 2 effectors, LDPA and LDPB, and the single deletions. And what I'm looking at is secondary branching again. And in the wild type, I'm seeing that expected reduction in secondary branching when we treat it with LCOs. And we also see that reduction in branching in the LDPB mutant. But suddenly in the double deletion and in the single deletion LDPA mutant, that response seems to be gone. This is telling me that LDPA is important for the response to LCOs and likely involved in LCO sensing. And I'm trying to be careful with the wording there, because this experiment is only saying that it's important for leading to the response, but not necessarily telling me that this is what's actually binding LCOs. But that's what I'm hypothesizing. I'm hypothesizing that this is the first thing that perceives an LCO right at the cell wall, and then relaying that information back into the cell, initiating the activation of ATFA. So To conclude, I've talked about how lipocytooligosaccharides are these regulatory molecules in fungi. And I've talked about how I discovered that an LDPA, a lysine effector, is important for LCO response in Aspergillus fumigatus. What I'm doing next is basically trying to confirm that LDPA is actually binding LCOs. And I'm doing this by purifying the protein and doing binding assays with collaborators. I've also talked about how I use Merlin to identify a transcription factor called ATFA, and basically validated that it is important for LCO response. And what I'm doing next is looking at the MAP kinase pathway and the two component system to basically ask, are they important too for LCO response? And I wanted to finish up by talking about how I uphold rigor and reproducibility. I basically wrote down the first 4 things that came to my mind when I thought about the words. And one of the first thing is I back up my data. I think one of the biggest things that scares me is that one day I will wake up and my data is gone. So I try to be very paranoid and try to back it up in as many places as possible. I also been very fortunate to get a lot of different mutant strains from a lot of different collaborators. But when I get them, I try to be very careful and I try to confirm that they're the actual mutant that I think they are. And I try to also confirm them sporadically. I also think it's really important to always do randomized sampling. I would do a lot of microscopy with the hyphae, and I always want to make sure that I'm not being biased by just selecting what I think should be selected. And lastly, I maintain an electronic notebook. It is a game changer. It's very easy to be transparent because you can just share it with other members of the lab, and then it's just very easy to update. But with that, I'd like to thank my 2 labs and my collaborators from the Roy Lab. And thank you. We'll take your questions. Yeah.
Nice talk, Chris. I had a question about, in the beginning, you said previously they thought that like only bacteria produce these molecules, but then like actually all of the proteins does it. How did they figure that out? Was that biomechanics? Something else?
Yeah, so initially we thought, so not only did we know that symbiotic bacteria would produce LCOs, we also knew that mycorrhizal fungi produce LCOs. So a member from our lab basically asked one day in the lab meeting, do other fungi produce LCOs? And that sort of initiated this entire project that he had where he's looking at different fungi and asking, do they produce LCOs? So it wasn't bioinformatics, it was Dr. Tomas Alan Rush basically looking at fungi and asking, do you have LCOs?
So actually growing them up and isolating them up for function. For a long time, we are using estimated for all your fungi and microbial fungi. We are using psychometry very and these are also not to define psychometry. But so much study, yes, but what about fungi over and just psychometry? Fungi is a weird one. Sorry, so the two of them, psychometry, but that's I want to for 10 years that our only over fungi was psychometry, and it comes out psychometry is one of the two that won't be different. What about when you wrote that I think yes, we haven't tried that because now anyway, but we tried some data and what we found from that is that using no, you can sorry, Zebra is not supposed to make IP. When you add LCO to Zebra stop, you the domain. So
Kevin?
I don't know if the studio might be focused or only on
Fastidiomyces were also tested, and they do. And they also do respond. So currently, the Dr. Thomas Allen Rush, he works with the Fastidiomyces and showed that it also regulates its own hyphal development. And then if there's no more questions, Kevin?
I just hit. So it's my pleasure to introduce Kevin Bao. Well, he's so Kevin joined my lab in 2020. It was a tough time to start learning a whole new system, as you know, but he did. Learned to work with Saccharomyces cerevisiae, the weird one, as well as some other species of yeast as well. And we work with those to do big experiments to kind of address long-standing fundamental questions in evolutionary genetics. And it's great that this is possible, but one of the challenges, of course, is that we have to make those experiments pretty large, and it takes a lot of effort over long periods of time to succeed at that. And so Kevin has found a way to motivate and inspire undergraduates and even high school students to help him with his research, which of course makes possible these larger experiments in my lab, as well as, of course, having the benefit of training the next generation of young scientists. So that's what I really appreciate about having Kevin in my lab, along with his really absurd sense of humor. Take it away.
Thank you. I hope everyone can hear me. I was a little odd. Never heard myself so clearly before. Hello, everyone. My name, as Dr. Sharper just said, is Kevin, and I'm going to talk about the fitness effects of mutations given a history of adaptation. Now, you think, Kevin, you work with yeast. Why is there a guy climbing a mountain? Well, we'll find out soon enough what this means. But let's talk a little bit. So our lab, I always say labs are sort of similar to, cartoon villains. They're really good at one thing. And the thing we're really interested in is what mutations do and how they affect evolution. Now, the thing about mutations is that they have a certain je ne sais quoi, right? Everyone's heard of them in some capacity or another. And for those who maybe don't know a lot about them, we think of them as being pretty bad. But for others who are maybe more on the evolutionary side of things, most mutations do nothing. And is the Zoom not working? Oh, I don't know how to do that. Thank you. No.
Which one is
Okay, wait, here, let me do it. I'll just, let me just go back into presenter view. Everyone look good? I don't know why it's.
Yeah, I think it.
Okay, great. Sorry about that. But for people who mutations generally have little impact on fitness, you can think of maybe a change in a non-coding sequence or a change that is synonymous. We have wobble base pairing. We have all these mutations that don't do anything to fitness as far as we know. If anyone says, I'm aware that there is literature that shows that even synonymous mutations have effects on fitness, but for the most part, mutations that do impact fitness, we see them as generally being deleterious. They're responsible for a variety of things, such as causing hereditary disease. They're responsible for cancer. We think many of these symptoms of aging are caused by the accumulation of mutations. And as you can see in sort of this model, most mutations, we believe, have very small impacts on fitness. And when they do have an impact, they tend to be negative. As a result, organisms generally try to have very accurate mechanisms for propagating their genes, and they don't actually have mutations super often. Now, previous work has shown that if you allow mutations to accumulate in the absence of selection, fitness does decline. This is from a study done on outcrossing nematodes. as you can see, over generations of MA or mutation accumulation, the overall fitness, by W here, declines over time. And this is a pattern that we've seen through experiments and through various different setups. However, that can't really be the whole story. There's no way every mutation is bad, because as every single intro biology textbook will tell you, mutations give rise to genetic variation, which is the raw material for evolution. And we know that over time, organisms change. After all, we're not lipospheres floating in a primordial ocean here. We are multicellular organisms, in part due to the genes that have evolved over time. Experiments in laboratory evolution show that organisms that seem pretty even well adapted to their environments improve their fitness over time when they're allowed to grow in the presence of selection. And of course, I wanted to show at least one fun image. So there's an Archaeopteryx, sort of one of those big models of the evolution of new traits. So we have evidence that mutations are beneficial, but how often are they beneficial and when are they beneficial? That's actually a little bit more debated than you might think. Certain experiments have actually previously shown that mutations may even get as close to being about 50-50 good and bad. Over on the left side of this, it's a little odd, it's inverted on this end. But on the side that you see, this study was conducted, it was a mutation accumulation study, just like the previous one that I showed, where selection was reduced through repeated bottlenecking in Arabidopsis thaliana. But what happened was, while the variance of the mutant lines increased, their fitness actually did not change. If you notice, the white triangle indicates the mean fitness of the ancestral lines. It's a little bit small, but you can actually, and it statistically showed in the paper that they weren't different. Their fitness wasn't declining. And their hypothesis was this was because about half of the mutations were good and half of them were deleterious. More recently, a study on mutant lines in Catamonas reinhardtii, which is an algae, also attempted to estimate the distribution of fitness effects and showed that there was sort of this symmetrical distribution across the center, about 0. Showing that about half the mutations they were modeling were good and half of them appeared to be deleterious. So what's up with that? Well, the hypothesis has long been that the fitness effects of mutations depend on the adaptiveness of the population, right? So if you look at the figure, if we take the cross to represent a population on a fitness landscape, and I'll go a little bit more into that, organisms who are higher on the fitness peak, they have a higher fitness. When you're at the top, there's nowhere to go but down. However, if you were lower down and you were taking random steps across this landscape, well, we predict that it is more likely for more of those steps to end up bringing you closer to the peak rather than necessarily further away. This hypothesis does make several assumptions. So first of all, many people, I'm sure here, have seen fitness landscapes, but we're actually dealing with one very, very particular type of fitness landscape. This is often called the Fisher's geometric model. It's a smooth fitness landscape with a single adaptive peak. I'm sure many of us here will say that doesn't seem super duper plausible, but the idea is that it's a good model for generally how evolution proceeds. And so under this model, again, right, if an organism starts out at the top of this singular peak, everywhere it goes down following mutation accumulation. And then if you have an organism that started off, again, along the slope here, well, we expect a more balanced profile of mutations. So what are you going to do? How are you going to measure this? So now is the chance for us to talk a little bit about the experiments that we do. But before I start about what we did, I gotta say, we put a lot of effort into trying to set up an experiment that was both rigorous and reproducible. So one great advantage of working with a model organism that grows in a test tube is that you can have a lot of tests. We could grow a lot of these at a time We could do a lot of things, but then they also reproduce quickly, which is really important to our experimental design. Additionally, whenever we could, we attempted to randomize the order in which we did things, and we removed human bias by having things be systematized. So instead of just going on like a plate and picking whatever column we wanted, we were very careful to pick them in a particular pattern, so there was no bias in terms of picking petites or ignoring them, or any sort of variation in that regard. Lastly, because as with any setup, there's going to be variation over days, over sections on your plate. We took a look at these block effects and we specifically analyzed them to see if there was an impact of these different block effects on the results that we got. And for the most part, we didn't have any block effects that actually affected our results. So our experiment required pretty much 3 steps to do. The first step was adaptation. In order to properly test our hypothesis, we needed evidence of adaptation. We wanted lives that had adapted to their environment. So we took Saccharomyces cerevisiae and we passaged them, allowing them to grow a large population size through these generations in novel environments. We formulated environments that were meant to be challenging and reduce their growth rate and have them adapt to it. We then performed mutagenesis. We used UV mutagenesis, we used mutants, and we measured, afterwards, we measured the fitness effects of those mutants in both the media that they had adapted to and media they had never encountered before. So, how do we set this up? Well, the first step was to set up the environments that they were going to adapt to. Our goal in setting up these environments, as you can see, was to give them a variety of stressors. We didn't want any environment to have just a single stressor, since previous literature showed that if you have only a single challenge, well, these tend to adapt in a pretty predictable way. And we didn't want all our mutations being in a single pathway or being along a very, very limited set of pathways. So each one was familiar with nothing that was, really acutely cytotoxic, that would just kill off all the yeast who didn't have a particular mutation. They were general stressors, hopefully similar to things that they don't have very, very particular pathways to adapt to. They have a mix of heavy metals, ions, and various other antimicrobial agents. We formulated these environments such that they showed a reduction in the growth rate when compared to YPD. That is the normal media that we grow yeast in. They really like growing in it. It's made from dead yeast. So they eat it and they grow. You are what you eat, so you become yeast. And it's non-restrictive, and all of our media are made from that YPD base in order to provide the nutrients, the carbon, nitrogen, but alongside these other stressors. We also tested each stressor individually to show that each one on its own had an effect on yeast growth. We also did tests with them in combination to show that they were not producing super huge synergistic effects or anything. So overall, they all had additive effects, which ultimately reduced the yeast to about similar levels of growth, as you can see over here. This process was repeated for a long time. I'll go over that in the next slide. But we would take the yeast, right? Each week they would be serially passaged. We would transfer from saturated culture about 100 microliters to new, fresh media each week. And then they were also regularly allowed to sporulate. What this did is that sporulation speeds up how quickly the yeast can adapt to environments, since we can more rapidly get, combinations of good or real together. We would freeze the yeast every week, and then we also measured the growth rate to see how adaptation was going. And then by the end of the process, we got a bunch of adapted yeasts. For anyone who's curious, each week we would use one of these fitness assays. They were stratified so that block effects, edge effect, which is a type of block effect, would be evenly distributed between our 3 groups. Because it was obviously the same setup every week, we also did analyze to see if particular well had any effects, and they didn't really have any major impact on the data that we collected. As you can also see here, the media ended up being actually sort of a little bit of a different color, in part because some of the metal ions do confer color to the media. This actually didn't affect our readings, since we were measuring optical density, and ours, as far as we could tell, they made no difference what color the media was. The yeast were allowed to grow for 42 weeks. This is actually a longer time because these are 42 weeks of growth. We didn't consider the weeks and actually formulating them to be growth weeks. So this took place over the span of about a little more than a year. And they grew for 42 weeks of adaptation. And by the end of the experiment, all of our lines of yeast showed substantial improvement over unadapted ancestors each week. Each week, not only would we assess the yeast that we are currently growing, we would also take our frozen ancestral yeast and grow it in the normal media to see how they did compared to our adapted yeast, which gives us a relative growth rate. As with anything over a long period of time, there was obviously a little bit of fluctuation. There were some weeks that look a little odd, but they did all return to normal, and those are usually due not to the adapted yeast having anything weird, but our ancestral yeast performing better than usual. So it's one of those things that happens over the course of an experiment, but I just wanted to point that out. So after 42 weeks, we felt pretty confident in that we could look at these yeast and perform the assay we wanted to do. We look at their fitness in both home and away environments and their mutants. But to do this, we wanted to really make sure that that was the case. So we performed a test for local adaptation. If people who do this in the wild would call these transplantation assays, where we took our different genotypes and we grew them in their home environment and compared it to how other genotypes grew in that environment as well. So here, A is in red, and you can see it's performing quite well, but a little bit of overlap with B, but it is substantially different. However, the issue we had was with environment C. Even though they seem to be doing better in our weekly growth rate measurement, they actually didn't seem to be performing better than both A and B adapted or even the ancestor in these assays. And we had a lot of replication in this particular assay. So we decided not to move forward with C because it simply didn't seem to have the signature of local adaptation we were looking for. So then we repeated this experiment with just environments A and B. And as you can see here, there was this clear crossing effect, right? Where A is better in environment A and B effectively is better in environment B. It should also be noted here that actually B adapted yeast in environment A and A adapted yeast in environment B don't actually perform significantly different from unadapted ancestral yeast, right? So with this signature of local adaptation found, we felt pretty comfortable moving forward, right? What we did next was perform UV mutagenesis and then assess their fitness. This took a little bit of work to do. What we want to do is analyze a variety of yeast and how they fare in both, again, their home environment, when they adapted to, and away environments, once they had not had a chance to adapt to. So what we did is we adapted yeast and we plated them out and we exposed them to fixed amounts of UV light. In order to make sure the UV mutagenesis actually worked, we also had control plates, which you would pick our control colonies from, that were put out the colonies, but were not actually grown with any UV exposure. We looked for a reduction in the number of colonies. This means that some of the yeast had, because they were all done from the same dilution, were killed by the UV. So the survivors presumably got some sort of mutations, sort of like a Cerevicia Mad Max situation. After that, we picked colonies. It's a little faint, especially on the projector, but we actually have a standardized system where we would take colonies from the center moving outward. We would take the colony, transfer it over to sorbitol, which is sort of just a osmotically neutral environment. We would send them down, separate them out, and put them in both environment A and environment B to be measured for their fitness. We repeated the same process, including the same colony selection process for our control. We went through all of the same treatment except for the UV radiation. They would even sit on the counter sort of away from the UV light, so that they underwent the same conditions, and we measured their fitness. This allowed us to measure 30-some mutant genotypes in 2 environments, for each particular strain that we did. So the results. Well, here is the results. I did A mutants home, B mutants home, A is away, B is away. We see that mutants, first of all, generally have high variance in their fitness. The red line on there indicates the mean of the controls who were not mutagenized. Now, this shows that we got obviously a range of fitness effects, but as you can see, it's not quite clear what it really all means. Well, actually, after further analysis, we actually found out that adaptive history actually does not seem to have an effect on the fitness effects of mutation. Really what we found is that mutations seem to generally be a little bit worse in environment B, and that it doesn't really matter whether you've adapted to environment A or not, right? And vice versa for environment B. So, well, now what? What does this all mean? Well, this could mean a couple of things. So first of all, Fisher's geometric model, right? The first smooth landscape. It simply has too many assumptions to properly model what is happening. The assumption that Fisher's model not only assumes a smooth landscape, it also means that every mutation has no epistatic effects, which probably isn't a realistic assumption. Indeed, more realistically, landscapes look like the ones in the middle and on the left here, where they are rugged, right? We have steps that can bring you up and bring you down. Instead of just being a single peak, there's numerous small local optima for adaptation. On the left there is sort of what is known as a max ruggedness, where there's no predictability about where your current fitness means for a mutation. I'm not going to make any claim as to which one is more true or which model particularly looks like this system. But perhaps it's more rugged than at least what Fisher's model, or too rugged for that model to represent what we have here. The other issue at hand may be that the adaptation wasn't complete, right? We saw the signature of local adaptation, but if you're sort of 40% up to the peak versus 10% up to the peak, we don't really have a good prediction for what the different institution of fitness effect would look like. And we know that adaptation takes millions of years to really go to completion. So perhaps we just didn't adapt them long enough, and given a genotype much more superbly adapted, perhaps it would look more similar to the results that we had expected. Indeed, these results actually aren't particularly surprising. There have actually been tests for the similar hypothesis done 2 other times as far as we know, once in Drosophila with mutations being mutations and looking at how they affected the genetic background, the environment, and a similar experiment with mutation accumulation and a transplantation experiment on Arabidopsis from France and Sweden. So both of those found results similar to ours in that it didn't really seem like you could predict the mutation based on adaptiveness. So this leaves us with a lot of future places to go. One thing that could be interesting is that we may want to look at the same mutation, but in haploids. Our current adaptive lines were diploids. We have recently confirmed through autophagy that they probably have to remain diploids, but some mutations effect might have been masked. And perhaps that's the reason for why we didn't see the effects that we expected to see, although I don't find it to be a particularly sort of likely scenario. It could be interesting to see what the recessive mutation that accumulated might have looked like. One more thing that we could do that again doesn't necessarily hit at answering our question, but could explain perhaps the pattern that we saw was to sequence our line and see what kind of mutations were generated. We used UW-Madison here rather than mutation accumulation, which generates spontaneous mutations, and we know that these mutations is somewhat skewed profile of mutation. So that could have affected our result. And so obviously the other step to do would be to re-perform this experiment using MA, which allows us to look at spontaneous mutations rather than UV mutagenesis, which generates a profile of mutations based on the mutagen that is used. So with that, I would like to thank our funding sources, the USDA Hatch Grant and the Genetics T32 Training Grant. Dr. Sharp, because you know, he puts up with all of my stuff. Denise Smith, who's our senior scientist, who in my many unbelievably helpful undergrad. Alexandra Brandt, who actually started off making the environment, and I wish I knew better why he took the things he did, because we have to take it over. But it's fine. Neil, really fantastic, super efficient. He does something that would take anyone else an hour to do in 10 minutes. Truly incredible, wonderful work. Aaron, fantastic guy, and Ian, one of my high schoolers who can help in the lab. So, you know, this couldn't have been done without all the people who helped. And I thank you all for listening to my talk. I know I spoke a little fast, so if anyone has questions, please. Yes.
I find this stuff really interesting, but it's outside of all from you. So can you help me understand sort of what you were predicting, what you thought would happen, like when you talked about, oh, you didn't get the outcome we expected. Help me understand like what you expected in the fall.
Sure, yeah. So I always explain that too. What we expected to see was that when we were well adapted to the environment, so the home basically, we would that's A home and B. Yeah, A adapted in A medium and B home is B adapted in B medium. We expected those things for the mutation to have a less deleterious business impact. Right? Because
you hypothesized that by adapting so long to the environment, they genetic robustness for growing.
Yeah, and so the mutations would be worse because you think that whatever genotype was made, you know, if you go in there and you slap the jackhammer, it would look a lot worse afterwards. Whereas in the away environment, we expected the mutations to be less.
Yeah, it's like your picture with the ball of it.
Yeah, exactly. Yeah.
Get more of the expected stuff because each environment has something that makes different home. And maybe that wasn't like, maybe that there is one set that was like off to one, but we have to do something family. So you can always have something that is that exactly but it's something for matter. So that could be an opportunity for
Yeah, actually, that is something that we had, that's 100% correct. That is something that we thought would happen. So if you had only a single pathway, let's say we adapted them to antimicrobial agents that, you know, prevent them from repairing their DNA or something. The issue with that is like, the effect would be what we expected. Where if you get a mutation in that particular pathway, it's bad. And if we don't get a mutation in that pathway, then it's sort of neutral. So we wanted to see what it's like having more sort of, I guess, natural looking environment with a variety of stuff. But yeah, that's definitely something.
one of these that like to be or these people with unhappy people or whatever, like maybe that case maybe
Yeah, no, we definitely thought about that, and there was work that was done at some point on temperature that was rescue, but this particular aspect was focused more on the raw language. But yeah, and sequencing might help us to look at what gene that we would be interested in.
Was this mine was that one is this one?
Good point.
This is only.
Yeah.
I'm wondering if this is just conceptual. But the way you draw the idea of adaptation, you draw it as heat.
Yeah.
So what is flatter and you know, robustness I don't know how this, I can't imagine how this would be necessarily selected for, but that over a period of time, you actually might be building, redundancy, right? I mean, it seems like it's not intuitive at all how that would happen, so you know, confused, and I am too. I'm just saying, you know, I mean, like, where, you know, we know that even in the uninteresting Escherichia, right, that there's lots of genes that at least in the risk conditions are completely, almost completely redundant.
Yeah.
You knock them both out.
You expect mutation to not be that.
That's what it says.
We'll have it down to bigger colonization.
Oh, maybe. I thought the condition was that we were, like, in one condition, in the home condition.
Yeah, but it's be bad.
We can use that everywhere.
Yeah, they're bad at it. And equally bad at all.
Equally bad.
Yeah, no, there are a lot of genes and a lot of pathways that could have evolved. We were hoping that this signature of local adaptation would show that they had some, unique adaptations. What hopefully would, I think, prevent us from having things where they were effective both amounts at the same time. If that's what you're saying, but I'm
No, but, well, I think from what May, I know you said, I think I was mixed up, so that's not a comment. Yeah.
Yes.
So how heterogeneous do you think these populations are? Like, I'm just trying to get a sense in my mind when you're thinking about working with these really large population sizes, and because you know that when you grow the culture as a whole, that it performs better. But I'm trying to get a sense of like what is the creative heterogeneity in the population, and then when you move to the stage where you are mutagenizing, then you are mutagenizing single clones, right? Yeah. And they themselves all have, they could all have single, they could have, if something did really well, they could all have really similar.
Yeah, every time we're actually currently investigating, so I did a little bit of analysis on the variances of control lines, root lines, and actually variance for the control line was higher than expected. That shows that there was a high amount of evidence in the population. And meanwhile, I will probably look at this.
Correlation conditions across the range, like the correlation conditions.
Yeah. It was about a week long process to sit in two correlation media for about a day. Both of them were 3 or 4 days. And they're all quite complex in their correlating. We're correlating pretty much every week that we started with, but they remain correlation complex. It's one of the using the book time is still there.
Did you go back to, did you after correlating, you go back to the growth?
Yes. Yeah, we're correlating.
Went back and forth.
Yeah. Question? Comment? We stopped at 42 weeks because it had been over a year. We wanted to sort of see what was happening. But they all showed substantial improvement to the slope from the beginning was greater than 0. They also were no longer showing the weird fluctuation in like growth rate that we were seeing. Again, that was mostly that 10 seconds, not from the adapting line. They didn't seem to be improving a ton at that point to stop. Maybe they plateaued out, maybe they had gotten to a bigger growth rate. Yeah, a part of I would love to continue doing this, but it's there's a lot of work to maintain all these and I have to keep going for who knows how long. But it will be easy to continue to do it. Especially with population C that we didn't see the evidence for. They are a little funky. They're always sort of weird at growing. So we'll have to see. That's our part.
So,
Thank you, thank you. I was,
Sorry, my questions were a little naive.
oh, no, they were,
Totally outside of my area. And it's just like,
they were great
I mean,
questions.
I work on Cervicia, but I'm not